teachers, and all distinguished attendees present here today. Lighting the lamp symbolizes wisdom, enlightenment, and the auspicious beginning of the program. It signifies the removal of darkness and spirit of knowledge. Goddess Saraswati is the goddess of knowledge, wisdom, and learning. Worshipping her is believed to bless the program with success and knowledge. Therefore, let us begin this auspicious session with the ritual of Deep Prajwalan and Saraswati Puja. I request respected dignitaries on the dais to kindly come forward and grace the lamp lighting and Saraswati Puja ceremony.
या कुन्देन्दु तुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डवन्दितकरा या श्वेतपद्मासना या ब्रह्माच्युत शङ्कर प्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा शुक्लां ब्रह्मविचारसारपरमा आद्यां जगद्व्यापिनीं वीणापुस्तकधारिणीमभयदां जाड्यान्धकारापहा हस्ते स्फाटिकमालिकां विदधतीं पद्मासने संस्थिता वन्दे तां परमेश्वरीं भगवतीं बुद्धिप्रदां शारदाम्.
Thank you so much, everyone. Before we proceed further, I humbly request all the respected dignitaries, teachers, students, attendees to please raise for the Maharashtra Geet. I request everyone to present here to continue stand for Savitribai Phule University with Thank you everyone. You may please take your seats for the session. Felicitation is a small gesture to express our respect and gratitude towards each other. I request Dr. R.S. Bhambar sir, Principal of SPH College of Pharmacy, Malegaon, to felicitate our chief guest, Honorable Dr. Harish Adke sir, Vice President of Mahatma Gandhi Vidya Mandir. As a mark of respect and appreciation, I request Dr. S.R. Tambe sir, Principal of Mahatma Gandhi Vidya Mandir, Pharmacy College, Nashik, to felicitate CDC Chairman, Honorable Rajesh Shinde sir. Deep respect, I request Dr. S.R. Tambe sir to felicitate our guest of honor, Honorable Dr. B.S. Yadav sir, Chief Executive Officer of Mahatma Gandhi Vidya Mandir. As a token of gratitude, I humbly request Dr. Mahalakshmi Mohan ma'am to felicitate Dr. P.N. Karkade sir, Principal of the Institute of Industrial and Pharmaceutical Technology, Nashik, operated by Adivasi Seva Samiti under Mahatma Gandhi Vidya Mandir. I humbly request Dr. Mahalakshmi Mohan ma'am to felicitate Dr. Sahil Mahajan sir, Sunil Mahajan sir, sorry. Thank you everyone. I request everyone to please have their seats. As we have gathered here today with curiosity and enthusiasm to learn about the topic cutting edge research and innovation, I would request our co-convener for National Workshop and Academic Supervisor, Dr. D.L. Athade ma'am, to shed light on the topic for this session.
Thank you, Erika. Greetings. A very good morning to all the respected dignitaries, delegates, faculty members, and dear students. On behalf of MGV's Pharmacy College, I warmly welcome you all to this two-day national workshop on cutting-edge research and innovation. The literature meaning for cutting edge means the sharp edge of blade, but in science, it refers to the most advanced and innovative technologies, and that helps us work faster and more efficiently. वास्तव में कहा गया है कि आज का नवाचार यानी नया विचार और कटिंग एज रिसर्च यानी अत्याधुनिक शोध यही कल की नई दवाओं, बेहतर उपचार और स्वस्थ समाज की दिशा तय करते हैं. In the present era, cutting edge research in pharmacy is being shaped by advanced technologies such as artificial intelligence, bioinformatics, and computational tools. These innovations are transforming drug discovery, chemical and phytochemical analysis, and the development of effective delivery system. This workshop is designed to introduce participants to modern research approaches that are shaping the future of pharmaceutical and herbal sciences. The objective of this initiative is to motivate the students, researchers to integrate modern technology with pharmaceutical and herbal and to create platform for sharing innovative ideas and promoting interdisciplinary collaboration. Today, Scientific progress is not only about discovery, but also about using advanced tools to achieve faster, more accurate, and impactful research outcomes. We are truly honored that distinguished and eminent speakers from across the nation will be sharing their knowledge and expertise in this event. In particular, Dr. Harun Patel, who has been listed 7 times among the top 2% scientists in the world by Stanford University, USA, and Dr. Shyam Prasad from Ayush CCRS, that is Central Council for Research in Ayurvedic Science, Kolkata, one of the premier government research organization in the field of Ayurveda, will be delivering expert session during this workshop. Through this event, delegates will gain valuable insights into cutting-edge techniques such as HPTLC, ICP-OES, that is inductively coupled plasma optical emission spectroscopy, and in silico tools. I hope this workshop will inspire young minds to explore new research directions and contribute to the sciences. I wish all enriching learning experience through this workshop. Thank you.
Thank you so much, ma'am. To enlighten and inspire us with wisdom and guidance, I humbly invite our respected principal, Dr. S.R. Tambe sir, to kindly address the students.
Good morning to one and all. The chief guest for today's workshop, Honorable Hariji Adke sir, guest of honor and the chairman of College Development Committee, Honorable Rajesh Shinde sir, the Senate member, and always the motivator for each one of us in the field of pharmacy, and my best friend, Dr. Bambar sir, the principal Katkade sir, the co-convener of the workshop, Dr. Nete sir, the nodal officer of the college, Dr. Mahalakshmi Mohan, the principal Mahajan sir, the chairman for morning session, Dr. Sandeep Sonone, the experts from the field, Gaurav sir and Monali Kado madam, all my faculty colleagues and the colleagues who are participating for this session, all the participants, it's my honor to welcome you all for this workshop, which will be there for next 2 days. There will be sessions, already ma'am has told you that the deliberations will be there that will enlighten us about what are the cutting edges which are happening right now in the field. And all these tools are official in the compendia. And really, it is important to enlighten us that how these techniques, they are important in day-to-day life. Yesterday, we have celebrated Women's Day. So many of the participants, girls are there, women are there. So I take this opportunity to wish you a very happy, healthy and fruitful Women's Day, because she is important. So how she is important that, and it is our responsibility also, that first is that socially it is our responsibility to keep them happy and safe. Next edge, that is always be honored when they are working in the field as a colleague or maybe in the various capacities. And always we should be the empathetic about our colleagues. So with these words, again, I welcome you all for this workshop. And this is the Quality Improvement Program which is sponsored by Savitribai Phule Pune University. So again, welcome you all. I conclude my talk. Thank you very much.
Thank you, sir, for your inspiring words and guidance. It greatly motivated all of us to strive for excellence and research. It is my great privilege to invite Dr. R.S. Bhambar, sir, to address the session with words of wisdom.
Honorable Vice President of our Mahatma Gandhi Vidya Mandir, Dr. Hariji Arke sir, the CDC chairman of this college and joint secretary of Adivasi Seva Samiti, Honorable Rajesh Shinde, CEO of our institute, our MGV Sanstha, Dr. Yadav sir, Principal Dr. Santoji Tambe, Principal Dr. Sunil Mahajan, Principal Katkade, key persons, speakers, or staff members, students. All of you know that the field of pharmacy is continuously evolving with new technologies, innovative research methodologies, and advanced analytical techniques. Today, pharmaceutical research is not limited to traditional discovery, but also includes modern approaches such as advanced analytical instrumentations, quality assurance, molecular research, and patient-oriented healthcare innovations. The main objective of this workshop is to provide a platform for the students, researchers, academicians, and industry professionals to gain knowledge about the latest advancement in pharmaceutical research and innovations. During these 2 days, participants will have the opportunity to learn about the modern analytical techniques such as HPLC, ICP, OES, lyophilization, and other advanced tools used in pharmaceutical research. Pharmacognosy plays a very important role in modern pharmaceutical research. It focuses on the study of natural drugs obtained from plants, microorganisms, and other natural sources. Even today, many life-saving medicines are derived from natural products. Pharmacognosy research helps in identifying bioactive compounds, standardizing herbal medicines, and developing safer and more effective drugs. We are fortunate to have distinguished resource persons from academia and industry who will share their valuable knowledge, expertise, and practical experiences with all the participants. Their guidance will certainly help young researchers and students to understand modern research trends and develop innovative thinking in the field of pharmacy. I am giving my best wishes for this 2 days national conference. Thank you. Thanks a lot. Good.
Thank you, sir, for your thoughtful address. It has set truly inspiring tone for academic gathering. I would humbly request our guest of honor, Dr. B.S. Yadav, sir, to kindly guide us.
Well, good morning to you all. Today's chief guest and Vice President of Mahatma Gandhi Vidya Mandir, respected Arke sir, Joint Secretary of Adivasi Seva Samiti, respected Shinde sir, Principal Bambar sir, Principal Tambe sir, Vice Principal Principal Kakade sir is here, and after a very long time I could see Principal Mahajan sir, who is occasionally present here. Mahalakshmi madam is here. Sonone sir, Kadu madam, who has come from a reputed institution to share her knowledge, staff members of the Pharmacy College, and my dear students. I find myself delighted, very happy for being with you all today morning for this inaugural function of 2 days national seminar on cutting edge research and innovation. For choosing or for selecting this topic for national seminars on which for these 2 days open discussions will be held. On this auspicious occasion, I will bring to the notice of all staff members, to the notice of my all students, that you all are aware that a tremendous development has been done nowadays in field of science, in technology, in healthcare, information. But thing is only that this is with the advancement of technology, that staff members should handle well, train their students well, make them skilled. That is actually the requirement of government, Ministry of Education, that whatever the projects, whatever the research students are doing, that should be innovative. So students should result-oriented projects, small projects should be given, and what I appreciate in every college, there should be an incubation center where the young minds should be nourished, they will be trained well for their future development. Because you all are aware that industrial development base of the GDP of any country. And country like India, industrial development is very near. That is why the government of India decided and asked AICTE to establish an incubation center under the Ministry of Education. And that incubation center actually asked every universities, research centers and colleges to get registered on the website of Ministry of Education Innovation Cell, and through that they actually provide financial assistance to the colleges for making the students aware about innovation, about incubation, about startup and entrepreneurship. And this is the time when the staff members should realize their responsibility
Further, it's my privilege to introduce our chairperson of the session, Dr. Sandeep Sonone sir, who is currently serving as a professor and head of the Department of the Regulatory Affairs at Meds Institute of Pharmacy, Nashik. Dr. Sonone has over 19 years of experience in teaching and research in pharmaceutical sciences. He completed his PhD in pharmaceutical sciences from Jawaharlal Nehru Technological University, Hyderabad, and he is also approved PhD guide at Savitribai Phule, Pune University. His research expertise includes quality by design, design of experiments, analytical method development, impurity analysis, and method validation. Under his guidance, more than 50 MPharm students have completed their research with 2 PhD scholars awarded and 7 currently pursuing their PhD. Dr. Sonone has contributed extensively to academic with more than 60 research publications, 3 books, and over 20 expert lectures at conferences, FDPS, and workshop. He also holds 2 patents, 2 design registrations, and a software copyright for the MET-ST80. With his vast experience in research, innovation, and mentorship, Dr. Sonone continues to inspire the pharmaceutical academic community. Sir, I request you to please come onto the gallery.
A very good morning to all of you. I welcome all of you for this 2 days national workshop on the cutting edge research and innovation in pharmacy, which is being sponsored by Savitribai Phule Pune University. Now we have the today's session that HPTLC and their role in a various with that the fields. So we say that we often hear that in the science that the simplest solutions is the most elegant one. Now everyone we know that for the decades we are knowing that the traditional TLC, which is what we say is that one of that the quick check method in the laboratory is one of that the simplest qualitative screening tool in the analysis what we say that. However, when we add the word HP, that is the high performance in this the HPTLC, it transforms it into the sophisticated instrumental method that is capable of the high resolution and we can say that precise quantitation. Now compared to the HPLC, what we can say that HPTLC it provides the parallel analysis that is more than 20 of that samples at time you can analyze with the help of that HPTLC on a single plate. Another thing is that visual evidences which is be not possible with that the HPLC which is be possible with the HPTLC it is be there. It's a very low cost or what you can say that low maintenance cost instrument compared to that the HPLC one. With respect to the applications, as we all aware, and sir is going to focus on all these the applications, but the major applications with respect to that the fingerprinting it is be there for a rare the botanical one, ensuring the stability of that any of that the life-saving pharmaceutical one. Detecting of that traces of that the contaminants in a food supply it is be there. So HPTLC plays a very important role. So to lead us this into the exploration, we have with us Mr. Thomre, who is from that the HPTLC expert. So sir is going to explore more regarding with this. So I formally welcome sir and I request sir to start with that the session. Thank you.
सो गुड मॉर्निंग टू एव्हरीबॉडी. मायसेल्फ गौरव ठोंबरे आणि मी इथे तुमच्या या वर्कशॉपमध्ये आय एम हिअर टू गाईड यू फॉर दी HPDLC टेक्निक. व्हॉट इज दिस टेक्निक अँड इट्स ॲप्लिकेशन इन वेरियस सेक्टर्स. ओके, सो this was the evolution that happened from PLC to HPDLC. In the earlier talks, you must have heard that this technique was earlier used at a very small scale as a preliminary test to check if all the tests are happening properly or not. So that is what was the basic technique that was then converted to a high performance analytical grade technique, which is able to give you reliable, reproducible results and even distinct results in some kinds of samples and some applications. Okay, so in 1960s this technique was introduced by the botanists. So these botanists, what they used to do is they used to extract plant juices, and from those plant juices they used to check for what all components are present in those juices. So from 1960s to 2000, TLC technique, as we know, different steps have been involved, right? So you apply your samples over a stationary phase, then that stationary phase gets developed into a mobile phase. After development, you go for the evaluations. Simply earlier the kind of evaluation was to capture the images, right? TLC being a visible chromatography or HPDLC being a visible chromatography, you are able to see the kind of separation that you achieve. Okay, so for all of these different steps, modules were being created by 2000. From 2000 to 2016, all of these modules were able to give you reliable results, reproducible results. So when they were giving you such results, these methods or those flow of analysis were being converted into files, or those were being documented as methods. As those methods were successfully being able to achieve the aim of the analyst, those were being created from 2016 to 2018. As these methods were used extensively in different sectors, it gained all of their regulatory compliances and guidelines for all of those different sectors. From 2018 to 2022, as you could see, one-stop solution for the HPDLC analysis came into existence, wherein unlike, like other chromatographic techniques, HPLC or GC, this technique got converted into a one-stop solution wherein you don't need to move for your stationary phase or your samples from your instrument. It was a one-stop solution wherein all of your samples and everything gets processed at a single go. So that was being done. So this is how the manual TLC got converted into a high-performance thin-layer chromatographic system. So this is the applicator used for application. Then you develop your plates using the automatic developer. Using the visualizer, you capture your plate images, and up above there you see the fluorescent image that is able to show you the kind of separation that you get and the presence of different components in your sample. Next is the scanner. So that scanner is able to scan your plate and it gives you the chromatograms. So using the chromatograms you are able to perform the evaluations. Next is the MS interface. The MS interface is a very, I would say it is a great tool which hyphenates HPDLC to mass spectrometer. Mass spectrometry is the ultimate powerful tool in the analytical field, as you all know. So it is able to extract components from your stationary phase which are separated by HPDLC and then know their identity by the mass spectrometer. So that is able to do that. Next, if you want some more information about your sample after development, you could derivatize your plate using the derivatizer. So that is how the derivatized plate looks like under white light. So all of these modules that you see are being controlled using the VisionCAD software, which is able to control all of these modules, and you could make use of these modules as per your will and wish, as per your parameters, to give you better results every time you perform the analysis. So this was the evolution that was happening between PLC to HPDLC. So the first kind of application pattern that was followed was through the capillary spotting, right? So these samples used to be applied in the form of a spot using the capillary, so that used to leach your stationary phase, right? And also the kind of separation that you get here in the TLC analysis is not good if you try to extract some data out of it, right? So that was what got converted to the high performance linear chromatographic results, giving you better sharper bands, and it is able to analyze more samples as per the traditional TLC analysis. Okay. So this was the kind of application thing that was being rectified, as the contact kind of application not giving you better separation was converted into spray-on kind of application using gas assistance, giving you better separation even at lower volumes and at higher volumes as well. So overall consuming less amount of sample and in a greener technique. So for these application modules are being used. So first is the Linomat 5 that is used for the sample application. Linomat 5 is a semi-automatic kind of a module which applies your samples in different formats. It could be in rectangles and different spots. And this is the stage over which the stationary phase is placed over the instrument, and your samples are applied over this stationary phase. So this stage is able to accommodate every size of plate, right from 5 by 5 to a 20 by 20 size plate, and the samples get applied through this syringe. So this syringe, it ensures that only a predefined volume of sample gets applied at your desired positions. Okay, and this instrument gets controlled using VisionCAD software. Next we have the automatic TLC sampler 4. So there you don't need to take, why this Linomat is a semi-automatic kind of a module is because the rinsing and filling of the syringe, it has to be done by the analyst there. So analyst has to be standing beside this instrument all the time while it's functioning. So you don't have to do that here in the ATS4, because what you got to do is just store your samples into the vials, and those vials get kept here into this vial rack. So automatically when you given command from the software, the particular sample gets applied to your desired position at defined volume. Also the rinsing and filling of the syringe happens automatically by these two solutions stored up over here into the instrument, which is able to rinse and clean the syringe after application. So this instrument, the ATS4, is able to accommodate 66 samples in a go. So you could have a huge sample capacity being accommodated there, and it is controlled using VisionCAD software. So with those modules, you could apply your samples in all these different formats based on the kind of application or the approach analysis that you are having. So if you want to go for quantitative analysis or quality analysis of multiple samples, you could go about this kind of application wherein you could have a sample being applied in duplicate in the first two tracks. Then in the third track, you could have your standard one being applied. Okay. Next, in the fourth and fifth track, you could have your sample 2, and in the sixth track you could have your standard 2 being applied. So likewise, you could have multiple standards being identified from a single sample, or multiple standards and multiple samples being identified from multiple samples. Okay, a single chromatographic go, that is just not possible in any other chromatographic techniques. Okay, so if you have some sample which is heavily loaded with fatty matrices, mostly this issue is being faced by toxicologists or forensic analysts. So what happens is, to eliminate those fatty matrices, means the main challenge why these fatty matrices are to be eliminated is because this HPDLC, being a chromatographic technique, it separates components. So during the separation, what happens is, at each separated position, these fatty matrices get deposited and they cause contamination when that particular component is to be evaluated. Okay, so as to eliminate those out, this same in situ cleanup kind of application has been followed, wherein the bands are applied at the height of 11 centimeters and then they are washed at the level of 12 centimeters using some like NXN. After development of that plate, after 12 centimeters, it is being cut off at 12 centimeters, and then it is inverted to 180 degrees. Once it is inverted 180 degrees, the samples are at the bottom edge of your plate, and now you could develop this plate into your desired mobile phase. So now you could achieve your desired separation pattern and perform your evaluations on without the interference of the fatty matrices. Next, if you wish to isolate something, or you wish to know its identity by some mass spectrometer or so, you could go about micro preparative kind of application wherein a huge volume and huge concentration of that particular substance can be deposited over your stationary phase, and after separation you could isolate that component as well. Generally, the kind of spiking that you do is using the premixes that you do, right? So HPDLC allows you to do that externally as well using the superimposition kind of application, wherein the standards expression could be checked in the samples. Okay. So this is the comparison between TLC and HPTLC with respect to the stationary phase. Stationary phase involves the silica gel which is coated over the plates that are used in the analysis. So here in the left hand side, if you see, this is a TLC plate of 20 by 20 size plate. Over this plate, only 8 samples are being analyzed, okay, and the kind of separation that I am getting is not that good, okay. And as I've consumed my whole size of plate, a 20 by 20 size plate, it would use a huge volume of mobile phase for its development, also would consume a whole lot of time for the development. So this being a 20 by 10 size plate, an exact half size plate of 20 by 20 HPTLC plate, it is able to analyze more samples, give me better separation, and automatically it consumes less time for development and giving you more results. So this is what happens as the miniaturization of solvents and everything takes place. So that kind of development happens using the development modules. So the manual kind of development happens using these twin trough chambers. So these twin trough chambers come in different sizes based on the plate sizes that you are analyzing. So during your chromatographic conditions, so as to create an inert atmosphere and to have a hassle-free development, these chambers come with steel or glass lids so that you could perform your chamber saturation as well. And next is you also get during your manual kind of development, what are challenges to be faces. We always need to stand over our plate and check if the mobile phase has reached to the solvent front or not. And also we need to check if the saturation time is being monitored properly or not. Even if there's some difference in the saturation time, it affects your development pattern. Also you need to have humidity control when you are having some complex sample or some inert sample to deal with. So as to take care of all these things, this automatic TLC development module is being used. So this is known as the ADC2. It is fully compliant with GMP, CLP and CGLP, and it is able to develop your plate with just 35 ml of mobile phase and get control over the humidity using the magnesium hypochlorite solution, monitor your solvent front using this laser assistance that happens here, and automatically precondition, activate and saturate your chamber during your chromatographic development. So this is the comparison between TLC and HPTLC. So in TLC what happens is you don't have any fixed plate size, okay. You could use any plate size, any combination of mobile phase and stationary phase is possible, and the data structure is not defined here in TLC. The photo images are being captured using the phone or your cameras, and there is no data orientation as such, right? So whereas HPTLC is having validated methods which are being highly standardized, giving you reproducible and reliable results every time you perform the analysis, and is compliant with all the latest regulatory guidelines and compliances that are available worldwide. Okay, so that is what sets HPTLC different from TLC. And the most important thing coming to the cost. So the running cost of HPTLC is incomparable with all such other chromatographic techniques. So it is very low per sample if you compare it with HPLC, and it is medium to high if you compare it with TLC analysis. Okay. So these are the features that you get from the HPTLC system. So you get standardized and validated methods, like I just said. All the modules that we have are software controlled. It is compliant with the regulatory guidelines that are the world is coming up. It ensures the activity and quantification of UV active components up to nanogram levels. For fluorescent active components, it goes up to picogram levels with a reproducibility of plus or minus 2% and is able to perform this high quantification and detection using the high accuracy and precision. Now this is a very important point that I would like to highlight here is multiple detections without repeating the chromatography. Now, in the previous slides only we saw the example of that, but I want you all to keep this point in your minds, and then we would see some examples related to this as well. Next is the easy MS hyphenation. It is not just MS hyphenation, but it makes easy MS hyphenation to the mass spectrometer. These are the different regulatory acceptances that CAMAG HPLC follows. So it uses this chapter USP 203 for identification and fingerprint of herbal samples. Uses this chapter USP 621 for scanning densitometry for quantification. Any kind of quantification that you do, so what would be the kind of approach? So it follows this chapter. Not only your essential oils, but even your fixed oils could be analyzed over HPLC. So how you shall go about it, that is being given there. Next is this USP chapter 2251. It talks about detection of synthetic APIs in herbal formulations. So these herbal formulations in our Indian subcontinent, what happens is they have a huge therapeutic window to show their therapeutic effect, right? So as to reduce their therapeutic window, these herbal formulations are being adulterated or spiked with synthetic APIs. So how you could detect those, that is what is given there in the USP chapter 2251. Next is the USP chapter 1064. It talks about parameter selection. So up above all these chapters, what are parameters would you select and what is the significance for selecting all those parameters is being given there. So it is compliant with, it follows all these different chapters from European Pharmacopoeia, Indian Pharmacopoeia, Chinese Pharmacopoeia and World Health Organization. So this is what the USP chapter 1064 talks about. It talks about the plate size that you would require for your analysis. Over that plate size, what would be the band length that you would apply and what would be the distance between them? What would be the development distance up till which the bands would be traveling your solvent front, in short? And do you need to maintain any humidity? Do you need to saturate your chamber before development of your plate? All those things are being given there. So these are the essentials or the perks that the QC lab gets from the CAMAG HPLC system. So up to 6 analysts could use a single HPLC system for their analysis individually. Okay, all these analysts, they have their own username and passwords, like we have our social media accounts. So nobody is able to hamper or create disturbance in your analysis. Okay. So this is a greener technique which just uses 35 ml of mobile phase for analyzing multiple samples up till 20. Okay, so it is a very quick technique that is able to give you results within just 90 minutes, right from sample preparation to your report generation thing. So these are the different regulatory compliances that CAMAG HPLC has. So you all being from pharmaceutical background, you all know what the significance of 21 CFR Part 11. Okay. Next, it is compliant with CGLP, CGMP. All the modules that we have in HPLC are IQ, OQ, and PQ qualified. Now, the thing that I just said, all the users have their own username and password to protect their works. And it ensures digital signatures up to 5 levels and user access controls up to 5 levels. It records all the audit trails for the system logs that get backed up, and it is compliant with the new regulations that are being coming up. So this is the stepwise practice of HPLC analysis. So first you prepare your samples. To those samples you apply those samples over your stationary phase, and then you develop them, capture their plate images after development, and perform your evaluations. Or if you wish to get more data, you could go for derivatization or could hyphenate this technique with some other techniques to get more data out of it. So it could be done using the mass spectrometer for the identity of the compound. For structural elucidation, you could go for IR-NMR, and you could also check for the bioactivity studies or toxicity studies using the bioactivity tests. So this is the stepwise sample preparation practice that we follow. So firstly, we weigh our samples. To that sample, we weigh the suitable, we add the suitable diluent. After adding the diluent, we sonicate our samples, the mixture. After sonicating, we centrifuge the sample. The supernatant that is being formed, that is collected for the sample application. So most of our samples are categorized into three main categories, as you could see. First being the raw materials, so the whole plant powders or root powders or your crude powders, those require just 100 mg per ml concentration for the analysis. When you subject these powders to some kind of extraction, you concentrate them, so 10 mg per ml concentration is required there. Next you have these standards, as them being in their purest form, just 1 mg per ml concentration is required there. So these are the three main components of HPDLC. So first is the mobile phase, which rises over your stationary phase and causes the separation. So just 35 ml of mobile phase is required here. And any combination of mobile stationary phase and mobile phase is possible as long as your method is not set. You don't need to degas your mobile phase unlike other chromatographic techniques. Next is the gas phase. So when you saturate your chamber using your mobile phase for 20 minutes before development, so that is what is the happens during the saturation step. That the vapors of your mobile phase, they create an inert atmosphere for your mobile phase to rise over your stationary phase, and also it gives you some control over the humidity then. If you wish to have excess humidity control up till 33%, that happens using the magnesium hypochlorite solution. Next is the stationary phase. So the commonly used stationary phase is silica gel 60F254. So this 60F254 is the dye that is added to the silica gel so that it makes the band fluorescent under the UV light. And next are this plate works in the normal kind of development. If you have a reverse phase kind of development wherein the stationary phase has to be less polar than the mobile phase, so that happens using the RP18 plates. And for separating huge components, you have cellulose fibers plates as well. So after development of your plates, you capture your plate images using the visualizer. So this visualizer, it is able to capture your plate images under different lights. So that includes white light, short UV 254 and long UV 366 light. Okay. So it is able to do the image enhancement thing as well, wherein your samples which are present in lower concentration, those could be intensified and seen. And it is also able to perform evaluations using those things. So it is controlled using VisionCat software, and these are the plate images that you get from the visualizer. After the image capturing, you go for evaluations using the scanner. So that detection happens using the scanner. So this scanner is the heart of HPDLC analysis. If you don't perform scanning, your analysis is same as TLC analysis, okay? So for performing your evaluations, this scanner is able to work in this huge range of 190 to 900 nanometers. In this range, it has these three lamps employed in it. So it has a deuterium lamp for UV region, it has a tungsten lamp for visible region, and mercury lamp for fluorescent zones, okay? So it is able to perform non-destructive kind of detection. So that is the best part about this instrument, and it is able to perform very sensitive detection. So this is how it works, and these are the different outcomes that you get from the scanner. So this is the integrated peak in 3D that you can see after scanning your plate. So this is the chromatogram that you get after scanning your track. This is the calibration curve that you plot when you are performing quantification. This is the multiple wavelength scan that you get when you scan your plate using multiple wavelengths. And this is the absorbance spectra. This absorbance spectra is a very important tool that helps in identification of components. How does it do that? This is how it does. So if you have, normally what happens is, if we have components separated at the same RF, we think that it could be the same substance, but that doesn't happen always. So as to know what is the main difference between those components, spectrum can be captured and the difference between them can be seen there easily. So this is another feature known as comparison viewer. So you could compare your old data to your existing data, or your today's data could be compared with the older data for different things. It could be for comparison of your images at different illuminations, or your profiles of your samples, or even the whole spectrum of the sample, or the particular band could be compared for different trials. So after development and detection, you derivatize your plate. So that happens using the derivatizer. So this derivatizer, what it does is it sprays a chemical reagent over your stationary phase. So these derivatizing reagents, what happens is they add a chromophore group to the developed components over your stationary phase, and based on their structures, they take up different colors, and those colors are seen after heating those chemical reagents. So that is being done using the plate heater here. With just 2 to 4 ml of reagents, your whole plate gets derivatized using some even dangerous derivatizing reagents which contain sulfuric acid and other such carcinogenic substances. So these are to ensure the safety of the environment and the analyst as well. So these spraying of the reagents happens inside the hood, so that gets adjusted, and these chemicals could be sprayed at different levels using the derivatizer. So this is what is the outcome that you get when you spray these chemical reagents over your stationary phase after development. So based on the different components present in a sample, different class of compounds could be detected. So things would be isolated and checked over the mass spectrometer using the TLC-MS interface. So this instrument ensures contamination-free illusion of zones from your stationary phase. So this is how it works. From a pump, the eluting solvent comes at a particular flow rate and dissolves the component over your stationary phase. And using the outlet tubing, the slurry is being sent to the mass spectrometer. But before going to the mass spectrometer, the slurry contains silica gel as well. So that gets filtered using the frit here. Okay. So how does it ensure that the 100% of your component has gone to the mass spectrometer? You could see the stationary phase after extraction of the zones. It is being punctured. Okay. And the only the component which was dissolved into the eluting solvent which goes to the mass spectrometer, its response is seen over the mass spectrometer in the MBZ ratio by. Okay. So this was the work that was done jointly by Shimadzu and Alcrom just to prove the speed of HPTLC along with mass spectrometer. So using the HPTLC system, some quinones and isoquinones were separated from teak tree sample, and some isoquinones were not known to the world back then. So those were isolated by the TLC-MS interface and were being analyzed by the Shimadzu LC-MS-MS. So from the LC-MS-MS library, those components were made known to the world back then. So all of this analysis just took about 2 hours or so. So with just 2 hours of analysis, some unknown component was made known to the world back then. So this work was presented at a ASMS-MS conference held at US, and was an award-winning paper back then. So as to check the bioactivity and toxicity of different components. So if you have some formulation you've designed, okay, and you wish to check for its bioactivity or check for its toxicity, so that happens using the bioluminizer. So this bioluminizer, what it does is it captures your plate images when you have subjected your plate to some kind of bacterial culture. Okay, now this bacterial culture is highly sensitive to minute toxicity as well. Okay, so any component that exhibits toxicity or shows milder toxicity or so from your sample, you would be able to know for its identity and for its position where it gets separated from your overall formulation, and what is the source of that particular substance which is causing toxicity and its identity. So that is what it does. So this is how your plate looks like when you've captured your plate image using the bioluminizer. So these dark zones, they indicate the presence of toxic components against this gray background. Okay, so these are the different kind of assays that could be performed using the bioluminizer, and to check for the different toxicity and activity of different components. So moving to the HPTLC Pro, this was the ultimate one-stop solution for the analysis. Okay, so the plate storage module is able to just keep your plates, so it is able to accommodate clean and processed plates as well. So your clean plates just need to be programmed from the plate storage module, and then from the application module to that particular plate, out of 5 you could have some set number has been applied at set positions. So it gives you this huge sample capacity of 75 samples at a time, and after application of those samples, when their plate goes to the development module into the desired mobile phase and the desired kind of development pattern, you could get it developed in isocritic as well as gradient mode. Unlike our older development modules, it doesn't have the TTC chamber or some other kind of chambers. It has Teflon chamber whose internal diameter is just about 3 to 4 mm. So again what it does is it converts the liquid state of your mobile phase into a mist or those vapors you could say, and with those vapors your stationary phase is developed, again making it even a greener technique for separation of components and getting maximum data out of it. Next is the derivatization. So the selection of the derivatizing reagent and rinsing and filling of the nozzle happens automatically. Next, it ensures the detection of components with an extended range up till 900 nanometers. Okay, so there it is able to measure the fluorescence as well as absorbance simultaneously. And next is the TLCMS interface. So this TLCMS interface is a kind of a different module wherein what happens is you need to keep in empty vials. So any zone that you wish to get eluted out from your stationary phase, you need to define its RF. So once you define its RF, that particular zone would be eluted out from the stationary phase and would be filled into that empty vial. Okay, and then that vial could be injected to your mass spectrometer, or you could do it as per your wish first. So next is the things that you go about method development of certain components. So first you check for the literature for that particular components. You check for its solubility. Next you prepare the samples for it, the kind of separation and the kind of preparation method that you go for. You select upon the stationary phase based on the polarity of the the nature of the component. Same goes for the mobile phase as well, which causes separation and isolation of that component. Next you work upon the concentration optimization, and then you define the detection parameters for those components. Once you've decided upon all these things, you go about validating that method. So that happens using these different parameters as per ICH Q2R2 guidelines. So that involves linearity and range, deciding upon the linearity and range, checking the LOD and LOQ, limit of detection, and quantification of that particular substance from your sample. What is the amount of accuracy that you could expect with that particular method? How precise is your method to detect that component? How specific is your method to detect that component from your sample? How robust is your method? If at all some chromatographic changes get happen when the development is happening, and most importantly system suitability test. If at all that analysis, that sample is suitable over that particular system or not. So just an overview about the HPTLC analysis and how the modules function, giving you the individual output. Moving to the applications. So to begin with, we have herbal applications. So there we could see the fingerprint analysis of bituminous red and yellow flask. So this shows the phytochemical changes that were happened for the flask for the budding stage and when it got matured. So the phytochemical changes were being studied here through fingerprint analysis. Next, this was comparative fingerprint analysis, wherein these individual components from the formulation were being checked for their presence in the formulation. So this is identification and quantification of lupeole in Celastrus paniculata. So this is how you go about quantitating a particular component from your sample. So this is simultaneous identification and quantification of multiple standards from Withenia sominifera, aerial and root plant. So this is an example of multiple detections without repeating the chromatography. So here you could see 4 standards are being quantitated from 2 different samples in a single go. So that is what is the efficiency of HPTLC. And one such example is identification and quantification of Satawarin. So all these herbs playing major role in our Indian economy. So these, to ensure their quality, to ensure their activity, these things could be analyzed using the HPTLC system, and fortunately it is able to ensure their activity and quality through this technique. So this is next example wherein identification and quantification of 4 markers from some gum sample is being done. So not just your plant parts, even your secondary metabolites, your gums could be analyzed to check different phytochemicals from it. Moving to the pharmaceutical applications. So this is detection and quantification of metafosine from different commercial samples. So this is an example of quantitative analysis for pharmaceutical samples. This is an example of again multiple detections for so this is the example of checking the limit assay, right? So even if you have some components whose label claim is to be checked, if that particular component is there in the set limit or not, so that gets automatically checked using the VisionCat software. Detection of impurity G in cyclophosphamide. Very important and a very crucial application that was developed over HPTLC back then, because the pharmaceutical industry was very much striving in the late 80s and 70s for the detection of these impurities. Who means back then nobody knew the source of these impurities, and nobody means no analytical technique was able to detect or quantify. So HPTLC was able to get a breakthrough on detecting these impurities and saving the world, formulations as well as humanity. Moving to the food applications. So this is identification and quantification of capsaicin from chili powder. So if you have really cheap or it is some sawdust that has been dyed using some color, that gets detected using the HPTLC system. This is the example of ensuring quality of saffron when it gets compared against the BRM, yani the botanical reference material. Another example of detection ensuring its quality by detection of its marker known as crocin from different samples of saffron that were being procured. This is again an example of limit test. So what happens is there are 2 kinds of cinnamon that are available worldwide. So these cinnamons get evaluated based on the coumarin content. Okay, so this coumarin content plays a vital role in the activity of the cinnamon, right? And this cinnamon or any herb that we consume, the activity of its active ingredient is what is the main essential thing, that is why we are consuming it, right? If it is there in an altered manner, there is no use of consuming it, right? So the right thing has to go in, so that gets checked using the HPTLC system. So this is another example of identification of rhodamine B in food products. So this is a very important application again, wherein this genotoxic, carcinogenic, and name it, that kind of culprit is this guy. So this gets detected from different food samples. So another example of detection and quantification of monosodium glutamate (MSG) or Ajinomoto from your different food samples, that happens here. Another is identification of sugars in honey samples. So here you could get your cellulose fibers plate being used here, because these monomers are huge in their size. So for separating them, these cellulose fibers plates are being used here so as to ensure the quality of honey that is being sold to you, or it is some sugar syrup that is sold to you. Another example of detection of synthetic dyes from different food samples. So next is an example of sunset yellow and tartrazine from tea powders. So this tea powders could be finished tea powders of sub quality, or this could be used tea powders which have been dyed with these two synthetic dyes and being sold back again into the market. So this is the identification of fixed oils. So not only your, like I said earlier, not only your essential oils, but even your fixed oils could be detected for their authenticity, for their activity, and for their adulteration as well. Because palm oil is the very cheaply available oil that gets adulterated in different useful oils. Another example is detection of mineral oil in pepper. So this black pepper, what happens is, it is to increase its weight. These peppers are being soaked in mineral oil so that they absorb this mineral oil, they get their weight increased, and automatically the vendor is able to give you means achieve higher weights in lesser quantity of black pepper, and even lower quality of black pepper. Moving to the cosmetic applications. So any toothpaste that claims to have eugenol in it, it could be analyzed and checked for its quality. Next, any cosmetic sample that claims to have curcuminoids in it, that could be checked for its quality. So sandalwood oil gets quantitated and detected. Moving to the forensic applications. So this is identification and quantification of cannabinoids in cannabis sativa. So any substance that is being seized over airports or some forensic sites, those could be checked for its identity, what is that component, or any person who's dosed with cannabis or is being suspected with some narcotic substances, though their visceral samples could be analyzed for the presence of different narcotic drugs. One such example is for cannabis sativa. Next, these are all the different narcotic drugs. So you name those drugs and those could be detected and quantitated using HPLC system. Not just your forensic drugs, but even your explosives. Our favorite RDX could also be analyzed and detected using Chemical HPLC system. These are the different references that you could use for your HPLC analysis. And this is how our lab looks like. This is how you could reach us, and thank you.
Now we have live session from their laboratory, Bulul. Okay. So we have a Zoom link.
Hello. So I request your patience, and here is now you'll have a live demonstration of the software and its feature with the latest features that are there, so that you could use these features for your analysis and research and get maximum data.
Yeah, hello and welcome everyone. So I hope you have got a very nice information from my colleague, our application chemist Gaurav. So today currently I'm showing you about the software, that how the software looks like, and what is other features or functions are there in this amazing software which is called as Vision Cats. And currently we have which is called as 4.2, which is a new upgraded Vision Kit software. So yeah, let's begin, and I will share my screen and let show you that how does the software basically looks like. And yeah, so I hope you can all see the screen of the software. So basically, this is how the interface of the software looks like. This is your Vision Guide 4.2. On the right hand side, you can see that some of the instruments are currently available in our lab. So as you can see, so some of the instruments have been connected and some of them are not connected. So some of the instruments you have been seen in your Okay, so you have seen some of the instruments like ATS, Denomet, EDC, visualizer, scanner, and this kind of other instrument. We have some of Pro also we have. So this is the overview of how the software looks like. So it's very simple to use whenever you get software, when you have get hands on this. So just you have to create new, you create a folder. After creating a folder, you will just get some of the application files. You have to create the method file. For the create method file, you just have to add the some of the instrument whichever you have available. As you can see, some of the instrument you added for the application purpose, like applicator, and it comes about the detection that we have to identify how the application has been worked, and then we have developed the plate, and then we did the image. The derivative is required as per the requirement. You can go for the derivative and then detect and then detection. For whatever wavelength you have to perform. As like that. So over here we have done some of the analysis, and let me show you some of the best file. So as you can see, when you open the application file over here, so as you can see, so when the application file is completed, you have to create this flag assignment, and these are some of the instruments. And after clicking the photos, pictures, so as you can see in the white image, you can see the caffeine and green tea has been performed, and on the different RF, you can just check out the spot amplifier and can change and identify this thing. You can even have option for the labeling of your work, as you can see for the application volume, how much you have been applied, and then after track number, whatever step you have with illumination and this is name, each and everything you can see on this single plate, and this is in 366. Even you get an RF tool, so you can mark the RF line, so where is your component standard is located, you can even identify like that. Even you can assign the tracks for the comparative. If you want to go for the comparison file, you can go with the track assignment like that. If you click the image in standard image, and just if you want to amplify the image and you want to see the better, more like enhanced kind of image, so you can even see like with the help of this one. You can even generate the profile. After generating profile, you can see some of the over here like that. So if you can see this is a complete 3D chromatogram. You can even move this your chromatogram. Currently we are using Steam with the help of this visualizer only. So like that, and there's come about the evaluation file. So in the definition you put whatever concentration you have added for the reference, that is your standard. Then the sample concentration you always put it out over here. Then you go for the integration work, and you can see as some of the wavelength is 280 somewhat, and you can check out some very nice chromatogram. As you can see, this is an increasing order, so which is your standard, and this is triplicates of your samples for the quantification purpose. And you go for the substance assignment. After assigning the substance, as you can see the green mark over here, so it has been assigned, and then you go for the calibration work, and after that you will find out the best CP. As you can see that it's so nice CP has been came off for 1.04%, which is very better. For the herbal it must be less than 5% somewhat, and like this you get your calibration result. And in result section you get your each and every kind of like that 280 nanometer, how much amount of sample is present, even though you can identify. As you can see in sample 1 in 10 milligram it was 153 microgram, and as same as with other also. Even though we have some of the other activities in the software and the optional, you can already go with the LOD LOQ. If you go with the LOD LOQ, and so we are currently seeing both LOD and LOQ, even though you can identify with that also. So in the result you can find out over here, you can see in the above. So LOD is showing 33.71 nanogram like that, and LOQ is 79.05 nanogram. So like even though you can identify what of that also, okay, how much the last detection limit would be go, and for the quantification of how much last till that you can go. And then you have a feature for the spectrum. So if you want to identify some of the compounds to check about that your sample. So these R is your reference. So this is a reference pick and this is a sample pick. So you correlate all the pick and you can see. So both the picks are completely collide and they are matching with each other properly. And when you click on the display maximus, so you get the wavelength of it, like what is the best wavelength. And there is an option for the spectrum library also, so when you can, if there is any phytochemical compound, so as per like if there is any compound you can see, so it will reflect over here. And this is the whereas basic work, like how the software looks like, and any other update would be Gaurav will tell you. And next about the new feature has been added in this, so will be continued by our business development officer Abhijit Kale. So we will speak about it. So over to you.
Yeah, thank you, Parth. Am I audible?
Yes, sir. Yes, sir.
Okay. Yeah, so as you saw, we have like received the recent major update in vision cuts now, which I'll be showing you. So is my screen visible?
Yes, sir.
Yeah. So now as we have already showed you that you can take an image in the software and go ahead with a number of applications regarding that or loading and so on and so forth. So this is one thing which I would like to show you is that this is one image for paracetamol, the daily, which is an OTC which is available all throughout. So this we had worked on it and
Yes, so the what just Parth was showing you, those were the general features wherein you are just able to get the data and it's not interpretation, say, but just detection of components, how you could find different results from your output that you got from different modules. Now, the feature that Mr. Abhijit Khare will be showing you, those features are something different which would help you interpret data using AI. And how that can be done and how one shall go about it, Mr. Khare will be showing us now.
Okay, Gaurav, thank you. Yeah, so coming back to what I was explaining, so there's a recent feature which has been added. So that is nothing but, in a layman's language, if I want to explain, that is your statistical analysis which you perform using different types of softwares or different types of maybe formulas or something which you perform outside the software. Then you create graphs, your charts, and your principal component analysis, and then different kinds of graphs, and then you present it. But this has been incorporated in the latest version of the software. So for that, what you need to do is, before you do any kind of an analysis, you need to determine as to what exactly is your aim. Like you want to find out the adulterants, or you want to find out maybe say some excipients, or maybe some impurities which are waiting for, or geographical variations if you are working with some herbals or something like that. So for this we had selected like paracetamol, which is used on a regular basis. So this is one impurity which is getting formed. So for that matter, different brands we have selected. So to continue with that particular analysis, we need to generate the profiles from the image and then export it to comparisons. So if you can see here, I have already opened one comparison file.
So what here the analysis was done for using the AI was, I'll tell you. So paracetamol tablet was analyzed for its presence. So paracetamol from different manufacturers. So it could be GSK, Cipla, XYZ. So all of them have the same paracetamol tablet, right? But the quality of the paracetamol differs for all these different manufacturers. So the difference between these paracetamol tablets being for the same concentration, same amount of API in the tablets, but still they had some variation. So we were to look for that variation. So for that, what we did is we analyzed all those different paracetamol samples. So if you see here, all these samples are for the paracetamol, different samples. This is the standard for paracetamol, and apart from these things that we are able to see, these were the impurities that were detected in these different samples. So not to go about the name, but these were some samples wherein some impurities were also seen. So what were those impurities? Based on those impurities, we tried to find the similarity or know the clustering of these different formulations. So that was done here using the AI's assistance. So normally, so as to know the variation between different things, अभी जो चीज़ होती है that is cluster, जिसमें के the statistical parameters are being used and the internal variation using TLS, all such different regressions you plot the variation that mostly statisticians know. हमें भी we being the from the scientific background हमें कुछ idea नहीं होता. Initially it all goes bouncer, but that is the aim of, I mean, that is the how you get the data out of it. Knowing the statistics is nothing there, but knowing how to achieve our aim is something what we are intended to do here. Okay, so when those things were being done, and the data evaluation sir would be showing you now. Yes.
Yeah, so as he explained, so the analysis was done, the profiles were generated, and from these stats, like the peaks and all was generated, and the data was input here in the software. So as you can see, there are different types of models and all which are available, which you can go into much details as and what the data type you want. So you can see, so based on the number of the bands which you see here, which are seen on the screen, accordingly different types of clusters were formed based on their closeness, and the difference between them is like how much data is taken in, and then the classification was done here based on different models like supervised and unsupervised. So preparations you can select here what wavelength and everything you want to select from 210 or 230 or 250 for which we had done here. So based on that we had got the data. Yeah. So I think that's some network issue. Yes. Pushing that and show here. So here you can see that cluster wise, as the cluster we saw here, these clusters 1, 2, 3, 4, 5, 6, 7, 8. Accordingly, the classification was given here based on like what is the cluster number, then what is the sample number based on the closeness and togetherness, and that's how the data you can interpret. So that's a short description of what the new AI feature is available in the software, and we'll hand over the session to Gaurav.
Thank you so much, sir. So that is how AI could be inculcated in our scientific field as well, wherein it helps us to generate data, not just to generate data, and it is also able to help us interpret the, though the components are looking similar to you. This generally happens in our Ayurvedic formulations or herbal formulations as well, because there is no standardization. So that using such tools you could achieve standardization in different formulations that we are coming up. Okay, so all the very best, and thank you everybody.
Thank you, sir, for your valuable guidance.
Okay, thank you, Mr. Gaurav, for a nice session. So the session is now open for the question and answer. Any query related to the instrumentation, the application part? Yes, ma'am.
Sir, how to know the system suitability parameters in HPTX?
So that happens. So the system suitability test, it happens by the detection of the component. So what you need to do is you need to specify that your standard or any substance that you are opting for system suitability test, you need to specify its position, at which wavelength is it seen, So generally what happens is at that wavelength when you are scanning, that particular component gets automatically detected. Excuse. So that is how it happens through the software. So when you detect that particular component at that particular wavelength, so it gets automatically detected. If doesn't get detected at that particular position, at that particular wavelength, system suitability test fails. So the system suitability test here in HPTC is little different, wherein it shows the authenticity of your analysis, wherein the standard that was being used was pretty stable, was pretty original, and was known to the sample and the analysis that was being performed there. So that is means once it gets detected at the specified RF and at the limit that you specified there, so it gets passed it on, and that is how it happens.
How to calculate the theoretical plates? Is it there is any provision to calculate the theoretical plates?
Theoretical plates.
It's from several systems suitability for propaganda.
Number of theoretical plates, I didn't get that.
Basically, in chromatography, we say that the number of the theoretical plates is a very important parameter to judge the peak. So it is be expected that the theoretical plate should be more than 2000.
Okay, so the area under the cover you must be asking about.
Not exactly the area, but this is depend upon that or what we can say that based on that the number of that the theoretical plates we can judge the broadness of that the peak.
Okay, so that happens means you would say the minimum concentration of that particular substance that has to be there, which gives you stable response, right? So it depends on substance to substance, but you could take about 4 nanograms is enough for a substance to give you better response.
Yes, sir. Thank you. Any other query?
So the bioactivity could be just detected, not quantitated. So again, quantification could be done based on means what happens is you need to scan your plate at a particular wavelength based on the response that you are getting. Okay, so you could take it as some color. Okay, so if it is some bluish color toxicity seen. Okay, so against a white background, what you could consider it is the substances which show toxic effect, those would be seen in bluish color. Okay, so as the intensity of blue color would increase, the toxicity would increase. Okay, in the same manner, you need to scan your plate at a complementary wavelength for blue color. So it could be 540 nanometers or 520 nanometers. So based on the difference in the absorption there, you could detect for the amount of toxicity or activity there.
Okay, thank you, sir. Any other query? Okay, so meanwhile I ask my query, sir. I also have one query.
Yes, sir.
Okay. In a HPLC, basically co-illusion it is being identified with the help of that the peak purity. So in a HPTLC, is there any provision that we can identify that co-illusion it is not there, and whatever that the band, the separated band it is there, which is be the pure one?
Yes, sir. We have this same feature as it is like HPLC thing. So the this peak purity kind of detection is not there when you scan your plate, but when you are capturing the spectrum, there is where you have this peak purity option. So what happens is the spectrum of that particular substance is captured at 3 different areas: at the start of that substance, in the middle of that substance, and at the end of that substance. So if there is no co-elution or addition or cross-contamination of the adjacent components present to our main analyte.
So with the help of that, the subtracting of that, the spectra, we can get with that the peak purity.
Yes.
Yes. Okay. Thank you, sir. So thank you very much, Gaurav sir. Sir has explained in a very detail about explained in very detail with the instrumentation and the applications of that the HPDLC. So starting with that the evolution of that the HPDLC, that is from that the 1960 that TLC how it is being used up to the 2022 that evolution of that the robotic HPDLC it is one. Sir has given a detail with respect to the instrumentation part, various of that the modules of that the HPDLC that we are using. Starts with that the applicator one, then derivatizer, then scanner. It is the MS interfaces where that sir has explained in a very detailed way. Sir has focused on that the various of that the application technique with respect to the quantitation. It is be there, in situ cleanup. It is be there, micropreparative. Or the superimposed type of that the technique. Sir has discussed various of that the development modules. Then sir has highlighted the features of that the HPDLC with respect to its sensitivity. It is be there, accuracy. It is be there, or the MS hyphenation. It is be there. Sir has highlighted that the HPDLC it is be one of that the regulatory accepted method in a USB, EP, IP as well as in that the WHO. Sir has explained various steps in detail, start from that the application to the scanning to the what you can say that the in situ applications one. One of that the new thing that I also learned that it is the bioactivity detector. Madam has asked about that the question. Then another features we have seen that the latest development in that the HPDLC, that is the HPDLC Pro modules. So automatically the application development, derivatization and the MS interfaces it is being done with the help of that the HPDLC. And further thing it is be the session it is being extended by that the applications work. So various of that the qualitative as well as that the quantitative applications in the various fields sir has discussed in a very detailed way. Further the session it is be we have seen that the live demonstration of that the vision with software. Mr. Parth Trivedi Abhishek Kale has given us oversight of that the use of that the HPDLC software, and also we learn about that how that the AI assistance can be used in the HPDLC development. So thank you very much, sir. Thank you.
Thank you, sir, for your valuable guidance and sharing your insightful knowledge with us. I now request the chairperson, Mr. Sandeep Sonone sir, to felicitate our speaker, Mr. Gaurav Romre, with the certificate. Thank you, sir. Now I request Dr. Swati Dalale, IIC coordinator, to come up on the dais and felicitate Mr. Sandeep Sonone sir. Thank you, sir. I further hand over the session to Ms. Asmita.
Good afternoon, one and all. I would like to invite the chairperson of this session, Dr. Dinesh Rishipathak, and the guest speaker of this session, Ms. Monali Kadu, to kindly come up the stage and take their seats. I request Dr. Manish Dunagade sir to felicitate the chairperson, Dr. Rishipathak sir. I request Dr. Anuja Bhosle ma'am to felicitate the guest speaker, Ms. Kadu ma'am. It is my pleasure to introduce our esteemed guest speaker for this session, Ms. Monali Sanjay Kadu, a dedicated researcher in the field of plant sciences and phytochemical research. She pursued Masters of Science in Botany from Mithibai College, Mumbai University, with a specialization in angiosperm taxonomy, graduating with distinction. She is currently pursuing her doctoral studies in botany at the Agarkar Research Institute, Pune, under Savitribai Phule, Pune University. Ms. Kadu has also qualified the prestigious national level examinations such as MHSET and GATE in life sciences, reflecting her strong academic competence and research aptitude. She has contributed to the scientific community through research publications and book chapters, particularly in the field of phytonanotechnology and plant bioactive compounds. She has actively participated in national and international scientific conferences where her presentation skills and research contributions have been recognized with the Best Oral Presenter Award at a national conference organized by Savitribai Phule, Pune University. She secured the first rank in her class while graduating with distinction in Bachelor of Science in Botany from Viva College of Science, Mumbai University. She is currently engaged in innovative research exploring the anti-cancer potential of plant-derived compounds, and a patent based on this work is presently under preparation. Ms. Kadu also possesses extensive expertise in several advanced laboratory and analytical techniques including HPTLC, TLC, LCMS, GCMS, phytochemical extraction methods, in vitro cell culture techniques, pharmacognostic standardization, plant taxonomic identification and authentication. Along with this, she is also highly experienced in research documentation, seminar presentation, data visualization and presentation. She has also contributed to academic mentoring, guiding and training students in their dissertation and research work. Her dedication to research and scientific advancement makes her an inspiring role model for students and young researchers. On behalf of our institution, I extend a warm welcome to Ms. Monali Sanjay Kadu. It is again a privilege to introduce the respected chairperson for this session, Dr. Dinesh Rishipathak sir, a distinguished professor and researcher in the field of pharmaceutical sciences. Dr. Rishipathak holds a PhD in pharmacy from Vinayaka Missions University, Tamil Nadu, and has completed his master's degree in pharmacy from Savitribai Phule, Pune University, with a specialization in pharmaceutical chemistry. With nearly 2 decades of teaching experience, he has made significant contributions to pharmaceutical education and research. He is a recognized research guide under Savitribai Phule, Pune University, and has guided numerous postgraduate and doctoral students. Dr. Rishipathak has an impressive record of over 45 research publications, authored books, and multiple patents, reflecting his dedication to academic excellence and innovation in pharmaceutical sciences. On behalf of our institution, I'm delighted to extend a warm welcome to Dr. Dinesh Rishipatak sir. Now I request the chairperson to kindly take over the session.
Good afternoon. Very nice introduction of guest and myself. Thank you so much, and thank you MGV faculty, principal sir, for giving us this opportunity. Now, HPTLC, as we all know, it is a rational technique for quantification of traditional medicines and other fields. Ms. Monali, her session should, can say, create interest in the prospective researchers from these sessions. I invite Ms. Monali to start his sessions.
Thank you, sir. Thank you, ma'am, for an elaborative introduction of mine. Thank you so much. Myself, Monali Kadu, and today I'll be presenting a lecture on HPTLC revolution. So, as you already know, Gaurav sir has already taken some of my part of the lecture. So, I'll just rush through those slides. So, let us begin. So before we begin, let me introduce what are the contents of my talk. So first, talking about the brief history of chromatography, followed by some of the extraction techniques. So in extraction technique, I'll be telling both the conventional method and also the modern method which is used for extraction. Next, I'll be talking on evolution from TLC to HPTLC and its advancements. Later, I'll be taking a detailed HPTLC fingerprinting and how it is useful in herbal identification or authentication. Next, I'll be talking on detection of adulterations in herbal materials, and then I'll be taking some of the detailed role of HPTLC, how it is useful in quality control of pharmaceuticals, and then I'll be concluding the session. So before I begin the session, please raise your hand if you have had tea or coffee today. Most of them have had tea or coffee, right? So when we have a tea and coffee, we just see it as a solution. It is like a brown or like a black color of the solution. And have you ever wondered what may be present in this? Mostly this question is for the master students sitting behind. So have you ever wondered what is present in the tea or coffee? Like we can just feel the bitter taste, right? Or bitter taste. Why is it so? Can anybody answer from that? This is because there are some of the complex compounds present in the tea or coffee, like the tannins, caffeine, and also polyphenols present in this. And here, many metabolites are invisible to our naked eyes, right? But the techniques, like analytical techniques like chromatography, which is very much important, and this will help us to understand what actual metabolites are present in the components like tea or coffee. And like tea and coffee is a very basic example, so I just start with that. And that is why we can use this technique like HPTLC to detect and separate the hidden compound present in it. So let us begin with the historical evolution of chromatography. Here you can see the scientists very tall columns are there. Earlier days it were like very tall columns were there. So how the chromatography has evolved from columns to also some of the paper chromatography techniques were used earlier. Then the column chromatography has advanced, like affinity chromatography were used, also ion exchange chromatography were used, and then how the TLC evolved, and then how the advancement in HPTLC took place. So let us see them one by one. So first column chromatography was developed by one of the scientists named Mikhail Svet. He was a Russian-Italian botanist and a biochemist. So because of this scientist, the column chromatography was first invented, and he invented it in 1903 for the first time. And from this scientist, the journey of chromatography has begun from 1903. So this is the illustration from one of his paper in 1906. So here this scientist has done, he has used adsorption column chromatography, which is packed with the calcium carbonate column, and in the figure 1 you can see he has used 5 different types of columns which is placed in a row, and somewhere here funnel shape glass these rods are present over here. So these are basically columns, and the eluates which will be get separated will be taken into this small flask over here. This pointer is not working. And the column B, you can see this is a little bit column which is for the larger sample size, and the diameter is also more than the earlier columns, and also the length of this column was more. So the scientists have separated the plant pigments using adsorption column chromatography. So he has separated the plant pigments like chlorophyll A, B, also xanthophylls were being separated by him. So next, after the column chromatography, paper chromatography came into existence around 1930s and 1940s, and this paper chromatography was based on partition between the stationary phase, where stationary phase was made up of paper or a filter paper or a cellulose paper, and the mobile phase liquid solvents were used. And from here you can see in this example the extracts were prepared, and it was loaded on a small size of cut filter papers, loading of the extract solvent system, and then it was developed by the mobile phase, and here are some of the visible spots you can see. So this method I had used mostly in my college days. Now I don't know this Gen Z generations, they must have used this paper chromatography or not, because nowadays there is like much more advanced chromatography present, like TLC, it is already coated on silica plates. So here one more example, it is a disc paper chromatography. So how many of you have seen this disc paper chromatography? So the basic principle over here is that wick is present in between, and this filter paper is placed on the beaker containing the solvent. So what will happen, the wick will absorb the solvents, and the solvents will move from the center towards the periphery, and this will produce the concentric rings of the compounds using this filter paper. So here is one of the photographs from the Russian scientist. He is, you can see how lengthy this paper is. Like he is observing the paper chromatography in this, and you can see how much tedious this process was earlier days, like using this chromatography technique. The next one is the advancement was partition chromatography. Here in 1930s and 1940s, the partition chromatography came into existence, and here mostly the separation was based upon the liquid these phases were there. And for introducing this concept of partition chromatography, there were 2 scientists who won the Nobel Prize in chemistry. So the Sir Martin, and also one of the Sir is Richard. Here they have won the Nobel Prize in chemistry in 1952 for introducing this liquid separation technique. So next we move on to the ion exchange chromatography. So ion exchange chromatography, after the advancement of other chromatography, this has also been laid, and mostly the charged stationary phase are used, and this charged stationary phase are used to separate some of the common compounds like proteins, amino acids. So here in this figure I have given one example of ion exchange chromatography. Here you can see a mixture of positive and negative charged proteins are passed through the negatively charged beads, and obviously the negatively charged proteins will repel this negatively charged bead columns, and that is why it will pass down, whereas the positively charged protein will bind to this beads, negatively charged bead, and it is retained back in the column. So next chromatography technique was adsorption chromatography. Adsorption chromatography, it is also type of a column chromatography only, and the major principle of this chromatography was the compounds are separated based upon the differential absorption. So here the solid surfaces were used in the column, and here the silica gel and the aluminium was used, and this is what we are using routinely in our labs, like separation of the plant pigments, also for the isolation of phytochemicals and purification of some of the organic compounds. So after the adsorption chromatography came into existence, some of the scientists were thinking like what can be done to minimize the solvent use and also use the sample in limited concentration. So in 1950, some of the group of scientists worked and they came up with the thin layer chromatography here. And everybody has performed this thin layer chromatography, right, over here. I guess no one is there in this lab who has not performed. So it is a very basic technique and it is very simple to do in the labs. But only one drawback over here we can say is that we cannot quantify this like as HPTLC chromatography, right? So the basic concept is the separation occurs on the flat surface. Like the earlier days, the like column were present, but here the in the miniature form and the flat surface form, we can get the separation and beautifully the bands can be resolved properly. So here the principle is adsorption and partition based differential affinity due to the stationary phase and also the mobile phase. So capillary migration also is an important thing in thin layer chromatography over here, and the mobile phase moves by the capillary action through the stationary phase over here. And this is how the bands get resolved and the compound gets separated on the TLC plate. And one of the key parameters in the thin layer chromatography is the retention factor, that is the distance traveled by the compound and the distance traveled by the solvent front. So let us zoom into the silica coated TLC plate, what actually silica plate is made up of. So the plates are usually made up of silicon dioxide, and here you can see silica and oxygen atoms are present. Oxygens are present all over this inside the like across the silica atoms, and some of the hydroxyl groups are present over here. So hydroxyl groups are mostly polar over here obviously, and they will participate into dipole-dipole interaction. So let us begin with the working of the TLC. So I'll just show you a short video. Like when we place, I don't know why. It's not compatible. So let us begin with a short tutorial. So we'll see the role of the mobile phase here on the TLC. We'll be using different combination of the solvents here, like hexane and ethyl acetate. So this is a different combination of solvent system. Chamber 1 and chamber 2 both are present over here, and we'll just run the TLC plate in this setup. So here we have used like 50% of hexane and 50% of ethyl acetate. So we'll see how the band will get resolved. So compound A, red compound is the polar molecule, whereas the blue color is the non-polar molecule. So obviously the red compound over here will be retained back as the silica is polar, and that is why it will get retained back. Whereas other molecule, let's use a different combination over here. We can answer from back side as a master student. What will happen if we use 75% of the hexane and 25% of the ethyl acetate? Can anybody tell from back side? What is happening over here, what you see in the TLC plate 2, you can see the compounds are moving very fast with the solvent itself, and the red compound is it is not moving further, and it is mostly absorbed on the TLC plate. That is why this is also not good type of a system, we can say, because it is mostly very much polar solvent is used over here, and that is why this is not a good type of solvent. Another solvent we'll use, what will happen if extremely if we use the non-polar solvent and extremely one side will use a polar solvent. So you can see in the chamber 2, both the compounds are not getting separated. Apologies for some disturbances created. We have already spoken about the conventional method. Three methods I have already discussed. The first one was maceration, then percolation, and so let us start with the extraction technique, like just brief. Like first one was maceration, soaking the plant material in any solvent. Here the passive diffusion occurs. Second one is the percolation. Here the solvent is poured from top, and some of the plant material is kept in this column, and continuously the solvent is passed through this column, and here mostly the most concentrated extract will be obtained. The third one was the reflex extraction. Here the heating mantle is kept at the below at the round bottom flask, and also the condenser is kept on the top, and the plant material is kept in the round bottle flask itself, and will get more improved quality of the yield, and this will show a good banding patterns on the HPTLs. Next one is the soft slit extraction. I guess everybody must have used this soft slit extraction technique. So this technique mostly operates via reflex mechanism, and here in this figure you can see again this heating mantle is present over here, and the round bottom flask is heated with the solvent, and the solvent will get evaporated from here, and it will reach the extractor over here, and the condenser, what it will do, it will continuously condense this solvent, and once the solvent reaches to some level in the extractor, in the plant material is kept over here in the thimble. So thimble is nothing but the plant material is wrapped in the filter paper, and it is kept inside this extractor, and continuously the solvent will get precipitated over this plant extract, and once it reaches a certain volume, the siphon tube, from the siphon tube again all the solvent will pass back into the extractor, right? So here I have given one short video. This video is not playing over here. So this is a much more improved version of the soft slit extraction technique. Here we can see, so here, okay, the video has begun. So here the heat is given from below, and how you can see how the condenser condenses all the solvent, and then again from the siphon tube all the solvent has come back inside the round bottom flask. So this extract will be used for some more techniques like rotary evaporator. We can condense this extract, and we can use this in the HPTLs. So this third one is the continuous soft slit extraction technique. Here it is used, and here we'll get much more yield, and no need to wait for like one cycle or two cycles. Here repetitively many cycles are present over here, and continuously we'll get the higher yield, and our time is not wasted. So another technique which we are going to talk today is modern extraction technique. So we'll begin with microwave assisted extraction technique. So here what happens, it is a rapid heating technique. Here in this figure you can see instead of the heating mantle, here oven is used over here. So what oven will do is it will provide even heat to the round bottom flask over here, and the sample will get extracted again. So this will disrupt the plant cell, and the solvent will penetrate inside the plant matrix, and then it will release the target compound inside the solvents. The next one is the sonigation technique, ultrasound assisted extraction. The main principle over here is that some sonicated waves are produced, and it will produce some of the bubbles over there, and it will collapse the microscopic bubbles in the solvent, and this will lead to the disruption of the cell membrane of the. So here two types of the sonation we will see, that is UAE probe type. The same principle applies for both of them. Another one is the bar type. Bar type of the sonation is mostly used for HPTLC technique, and we'll get a high yielding profile of HPTLC over here. So main mechanism involved in this type of extraction is cavitation of the bubbles, and like very high pressure of the sonicated waves are produced over here to crack the plant tissues, and also the cell disruption and fragmentation occurs. The next one is the sonoporation technique. So formation of pores will occur in the cell membrane, and the metabolite will come out of it, and this will also give us the higher yield. Also it will reduce the time and solvent consumption, and also it will give us the high quality of the bands. Next one is the supercritical fluid extraction technique. This is an instrument for this, and here mostly we will be using carbon dioxide. Carbon dioxide, it acts as a, it will extract mostly the non-polar compounds over here. So carbon dioxide over here in this system behave like a gas-like diffusion due to the high pressure over here and also the high critical temperature. So we'll see mechanism over here, how the non-polar compounds are extracted over here. So most non-polar compounds which can be extracted using this technique is lipids, essential oil, terpenes, and some of the flavonoids can also be extracted, though they are polar substances. But by adding some of the enhancer over here in this carbon dioxide, we can also extract some of the flavonoids from here. So let us see the basic working of the this supercritical fluid extraction. Here you can see the reservoir of carbon dioxide is present. So the carbon dioxide present here is liquid form, and the carbon dioxide which is in liquid form will be pushed through this pump forward, and the carbon dioxide gets compressed over here, and it is passed through the heater over here. So the temperature will be more than the critical point over here, and this will produce the supercritical carbon dioxide. So this carbon dioxide will be pushed further into the extractor over here. So extractor basically consists of plant material of our interest over here, and the carbon dioxide along with some of the active metabolites will be passed through this pump, and it will over here this point the carbon dioxide, the pressure will be lowered, and that is why the extracts or the metabolites of the plant will get precipitated over here, and we can collect this from the outlet of present over here. Also the carbon dioxide which was remained over here will be pushed back from the gaseous state to the liquid carbon dioxide phase in the reservoir again. So there is no loss of carbon dioxide over here, and it is environmental friendly. So next one is the pressurized liquid extraction technique. So this technique mostly evolved again the high pressure and temperature. So difference between this is from the supercritical fluid extraction. What is the major difference over here is that we can extract both the polar and also the non-polar metabolites over here. So here is the basic working of pressurized liquid extractor. Here you can see all the different type of solvents are kept, A, B, C and D. So this is both the polar and the non-polar combination of the solvents can be used, and this solvents are pushed through this pump over here, and then there is a nitrogen gas in between here. So it is connected to the purge well. So whatever air which is not required will be vented out from here, whereas the pressure will be maintained by using this nitrogen gas, and using this pump the solvent will be pushed towards this extracting cell over here. So extracting cell, one is present over here, so heating will occur over here, and also some plant material are present already over here, and this will extract out all the metabolites from the plant, and this compounds will be collected into the collection vial over here, and then the waste vial is present over here. So all the unwanted solvent will be separated over here, and the air will be vented out from here. So this sample we can use for performing chromatography or any other techniques. So we'll see how the effect of extraction method on HPTLC fingerprint effect. So if we take two plants from the, if we take one plant from different source actually, what will be the effect of that on the fingerprinting? Will we get the same fingerprinting or will we get a different fingerprinting? So the fingerprinting will vary over here because the extraction technique will be different over here. That's why it is like different. So we'll see over here how like expected HPTLC fingerprint is seen by using the extraction method, by different extraction method. So maceration, as I mentioned earlier, you will see just a fewer bands over here because this technique is not much more efficient to drag all the compounds from the it. The soft light extraction, ultrasound or other modern techniques, it will yield much more stronger quality of the bands on the HPTLC plate because obviously the heat and the temperature and the high pressure, it will disrupt all the plant cells, and that is why we will get the high intensity of bands in the much more advanced technique. So here I have mentioned some of the example of the extraction method, and here I am taking some example by using methanol, how the fingerprinting is seen. So by using methanol extraction, most of the flavonoid will be extracted over here. So here you can see yellow color of the band mostly, and by using the solvent methanol over here. Next one is the hexane extraction. Non-polar solvent will extract mostly the oils or maybe lipids on the TLC over here, and mostly the band can be seen a blackish or brownish in color. Next example is for ultrasound extraction. So here is a fingerprint for the same, and here you can see higher metabolite intensity over here. Same we can see for the microwave extraction technique also, because both this technique are will give higher yield, and the resolution of the fingerprint is much more higher than the other techniques. So if we use the same plant and different extraction technique, how it will affect? It will affect the fingerprinting pattern obviously, because the extraction needs to be standardized before performing the HPTLC actually. So the key impacts of extraction is that metabolite composition will vary based upon the extraction technique over here. So different solvent will extract the different type of the phytochemicals. Also the band pattern will vary if we use the different extraction technique. Also the reproducibility of fingerprint is different, sensitivity and also the selective enrichment of the extraction technique will vary. So next we will see the HPTLC workflow or methodology. So I will be taking one example over here, that is piper nigrum, that is black pepper. So it is first collected from the field, or maybe after that we can dry this sample properly, and then the dried sample is pulverized and it is made into powder by using a mechanical grinder or maybe using a mortar and pestle. Then we need to dissolve 1 gram of any of the plant into the 10 ml of the solvent. So this is the standard protocol usually. So it will be more concentrated extract will be formed. So this extract can be then extracted using different type of the extraction method over here. And then the sample is then centrifuged for around 10 minutes, and the filtration of the sample is very much essential, so that we'll get a proper bands and it shouldn't interfere in the syringe as well in the HPTLC. So filtration is very much important to get the uniform metabolites and the binding patterns. So next we come to the HPTLC methodology is first we'll need to fill all the data in the software as shown by the Parth sir earlier in the video. So after filling all the parameters, we'll fill all the plate parameters, automatic development chamber parameters, how much solvent should be used, what solvent should be used, how much time it should be run, like 70 mm basically the plate should be run, and everything we will enter in this software. After that we'll apply the sample by using a sample applicator. So here in this image you can see Limonade 5 type of sample applicator, and the plate, TLC plate is placed on the stage over here, and then we will spot all the bands on this by using a sample applicator. And once we finish with the spotting of all the bands over here, we will develop a chromatogram using ADC chamber over here by using 25% of the saturation mobile phase, and 10 ml will be put on the another inlet over here for development of the mobile phase. So once the chromatogram is developed, it will be detected under the UV visualizer, or if it is visible in the UV it is well and good, otherwise we need to derivatize sometimes, or sometimes we use UV or mercury lamp to see this detection. So here in the visible light you can see, as I have taken the example of Piper, you can see some of the bands are visible clearly over here. That is a piperine band seen over here. But for more clarity, we have visualized it under the UV light, and you can see much more clear banding patterns over here. So two types of marker were used over here. First one was piperine. So here piperine was used in lane number 1, and the second Borneal standard, which is present in Piper diagram, was used. So you can see Borneal is also present in the other lane. The lane 3 to 11 is authentic samples. So we can conclude that if both of these are present, the sample is genuine, and it is also not a degraded quality, and thus we can authenticate the plant material. So after understanding the qualitative analysis, we'll move to the quantitative analysis. So for quantitative analysis, we will be using TLC scanner. So we'll scan the plate accordingly to quantify the band of interest. For example, here I have taken an example of piperine. So here the particular band of piperine was selected, and then it was scanned at different wavelength. If we don't know any wavelength of the standard, we need to scan it into wide range, that is 190 to 800 nanometer around. So all the lamps will be covered over here. So different wavelengths will be covered. So here UV, for scanning at UV light, deuterium lamp will be utilized. For fluorescence, mercury lamp will be utilized, and for scanning the plate at visible light, after derivatization, we will be using a tungsten light lamp over here. So here is the piperine wavelength, we have got it at 342 nanometer, and this has been shown by the scanner over here in this image. Here you can see piperine was present as a standard marker. So Borneal tracks were off, so it is not showing over here. So we can just select whichever bands we want to see in our plant. So next we can see black pepper, it is present. So piperine is present in the same RF value as in the sample of black pepper. So we can conclude that qualitatively and quantitatively and qualitatively both, like piperine was present in our sample and the sample was genuine. Again we have calibrated this sample further, and then we have quantified, and we could get the yield of this much nanogram at the 342 nanometer. So next, derivatizing agents. If the samples are not visible on our plate, we have to use some derivatizing reagents to make them visible. So mostly different class of metabolites will be seen by using a different derivatizing reagent. For example, flavonoids, for detecting flavonoids, mostly it is like yellow orange in color when it is derivatized with NP-PEG reagent over here. Another example I can take of alkaloid, it is derivatized with dragon drop reagent. It will show orange to reddish brown color and so on. So others are mentioned over here. So the main conclusion is that different derivatizing agent will show different band colors, and for it is specific for different metabolite class. So we'll be seeing now how HPDLC is used in quality control and detection of adulteration. So mostly I'll take example of very common this papaya seeds. It is piper nigrum seeds are adulterated with papaya seeds mostly in the market to get the economic profits. And also another image is of saffron. The saffron mostly the stigma part is used for and in our cuisines or whatever sweet dishes what we use. But the stigma, it is like very much costly. It is like 1 kg for this stigma of the saffron will cost around 3 lakh rupees. That is why to increase the economic profit, some of the traders will adulterate it with similar looking of the compound, like some safflower petals, like which are orangish in color. They will put the petals which are dry form. It will look like this. Or sometimes some threads are also colored and put inside that, and that is how they gain the profit in the market. So HPDLC role over here is to detect the adulteration. So we'll be taking 1 or 2 examples over here. So for example, palm oil example I'll be taking, like it is very cheap palm oil. Actually, it is available at very low cost, and that is why again the trader will adulterate it with cheaper quality of palm oil. So, what is the harm of using the palm oil in other vegetable oils which we use in our daily preparations? So, can anybody see the oil and tell us that whether it is adulterated or not? We cannot identify it with our naked eye, right? So, palm oil, what it contains mostly 50% of saturated fats. That is why a major health concern can be raised, like some risk of cardiovascular disease can be seen because of the mixing of the palm oil in our regular oil. So this is some of the method overview of detecting the palm oil in the vegetable oil. So here oil, it was diluted as it is non-polar, it is diluted with hexane or chloroform. So this is the method overview. We'll take an example quickly. So here is the main objective of this running this TLC plate was to detect the palm oil from the other vegetable oil. So in this image what you can see is palm oil. This is a standard palm oil over here. In second and third lane the palm oil of different brand was used. So and other lane number 4 to 11 consist of other regular oil which we are using in our food preparation, coconut oil, groundnut oil, sunflower oil, etc. So here you can see in the first lane at RF 0.18 around, and here also you can see somewhere like unique bands are present in the palm oil which are present more pronouncedly in this other oil. But by spiking it, we have found that the oil was present in other this vegetable oil also, and it is like, and thus the authenticate it was authentic or not we could just prove by HPDLC. So that we can use HPDLC for the quality control in the food and pharmaceutical industries. Next one, we have taken example of monosodium glutamate. It is normally known as Ajinomoto, and this is used in various cuisines. Also, it is used to enhance the flavors of the food, like noodles, pastas, or other packed products. Also, the meats and other things. So earlier we used to think that it is very much harmful for us, but recently, like FDA has told that it is safe for consumption if it is taken in limited amount. But though there are some of the cases like short-term symptoms are available by taking excessive other like outside food like containing MSGs. So symptoms are like headaches or like redness in chest and neck, etc. will be there if we use a pronounced consumption of this MSGs. So here, how can we detect the MSG in commercially available food products using HPTLC? So here we have detected it by using a different kind of derivatizing agent, anhydrine reagent over here. So here in this plate you can see clearly, here you can see red band, you can see red box, it is MSG standard marker, whereas yellow one is present in the food materials which was detected. So this is how the HPTLC can be used to detect the adulteration. The next one is melamine example. Melamine, what is melamine? It is usually three like amino acids are attached over here. 3 amino acids are attached in this compound mostly, and this is added in milk products mostly to increase the protein content in the milk. And mostly it is used by some of the fraudulent producers, and it will thus create some risk like kidney stone and renal failures. So if the milk which we are having is safe, so mostly we have tea, coffee in our day-to-day life. Also we have other fruit juices, etc. So it is a public health concern if melamine is present in the milk and its products. So study has found that many milk was being adulterated, and by raiding many of the milk producers, they were caught because it was adulterated. Also the Operation White Poison has in 2020 recently in Pandharpur, like some of the milk producers were caught because of the use of melanin was there. Again other chemicals were also present with them, with this fraudulent producers of milk. And around they have claimed, like some investigators have claimed, like 50 liters of more milk could have been produced by using those chemicals present with this fraudulent producers. So how we can detect the melamine in the milk using HPLC? So this is the sample preparation method over here, and here we have detected the presence of melamine. In this image you can see. So basically I'll be taking a last example. Sudan red dyes in the chili powder. So mostly illegally Sudan dyes are added to increase the bright red color of the chili powders, or maybe to improve the market appeal, also to compensate the poor quality or aged spices, etc. So main concern of using Sudan dyes was a potential, it is a potential carcinogen. Mostly in Europe in 2005 around, like many companies were closed because of use of Sudan red dyes in their products like sauces and also some chili powders. So here is a method overview of what we have used. And here is the detection of Sudan red dyes in chili powder using HPTLC. So we have taken in the lane number 2 to 7, they are genuine chili powders which are present in the market, and we couldn't find any bands over here, right? So we have tried spiking it with this Sudan red dyes over here. Like Sudan red dyes are 1, 2, 3, 4 different types of Sudan red dyes are used, and by spiking also we couldn't find any Sudan red dyes present. So that is how we confirmed that the chili powder which we have spotted over here were genuine. So why use HPTLC? HPTLC is much more sophisticated instrument than other techniques like HPLC and LCMS also, and sample analyzed is much more higher than HPLC and LCMS. In HPLC or LCMS, only one sample can be analyzed at a time. Sequential batch analysis is done, like one sample is run at a time over here, and that is why it is like time consuming process. So cost of analysis was very much low by using HPTLC than other techniques. Also, the visualization is direct visualization of band. Here we cannot see visualization of bands. Also, the suitability of the herbal extract is excellent. We can just visualize by using a fingerprinting profile. And it is also solvent consumption was low as compared to other methods. So that is why HPTLC can be used. So major application of HPTLC, sir has already covered that. So I'll just highlight the application. It is used in plant authentication. It is also used in the quality control, food analysis. Also, it is used in forensic toxicology and cosmetics, whereas it is also used in environmental analysis. So, moving on to the conclusion. So, what we have seen today is TLC and its evolution from like chromatography, different type of chromatography we have seen, like column chromatography, right from the Michael Sweat scientist. He has introduced column chromatography in 1903, and then we have moved on to the advancement of HPTLC. Next, we have seen the two different types of extraction technique, both the modern and the traditional technique which was used, and we have seen different types of the extraction technique within it. Also, we have seen how HPTLC can be used as a quantitative analytical tool in pharmaceutical industries and chemistry labs. Also, how the fingerprinting is important to authenticate the herbal plant and also to detect the adulteration patterns. And lastly, we have seen of HPTLC, and we can say that it is a very cost-effective process, and it is rapid, and it is used for quality control of the drug substances and formulations. Thank you. Any questions?
Thank you for this nice session. Any questions from the audience? Yes. I think no question. But one question from my side. I'm from the synthetic chemistry background. Can you highlight the applications of this HPTLC synthetic chemistry? Okay. Any clue?
It can be used in cosmetics also.
Okay.
Chemistry also. For cosmetics, there we can use it as like maybe lipstick or something. It may contain some of the harmful chemicals which will be interfering with the human health. So we can detect harmful chemicals in cosmetics also.
Thank you. So I'll conclude this session. Madam has nicely covered the history of chromatography, including conventional techniques, and then we have two HPTLC techniques. Different extraction methods, because extraction methods, there is a difference in the fingerprinting pattern. She has explained very well. Then she has explained how to detect the adulteration in various samples, quality control of herbal products, and various applications in the field of like the milk adulteration, oil adulteration, the very like MSG adulteration. Then why we have to prefer HPTLC with respect to the other technique like HPLC and other with respect to cost, time and solvent consumptions. So with this note, I conclude this session. Thank you. Thank you all the audience and thank you.
I request the chairman, Dr. Rishi Pathak sir, to kindly felicitate the guest speaker, Ms. Karuna. I request Dr. Daksha Atharde ma'am to kindly felicitate the chairperson, Dr. Rishi Pathak sir. I request everyone to kindly proceed for lunch in classroom number 5 on the second floor of the building beside this one. Thank you.